Thank you very much for your very kind introduction. And yeah, very much appreciate the kind invitation from Professor Ramirez and also the SCB. Yeah, so let me share my screen. Yeah, so the title of my talk is From DNA to UI, Connecting the Dots Between Theory and Application in Bioinformatics. Today we're gonna pretty much talk about how, like in this development stage where we have problems from biology and then how we could, transcend that into an interactive data app where biologists, chemists, or other researchers could have easy access to the underlying model. Yeah, so as already mentioned, you could also check out my Data Professor YouTube channel where I create like educational content all for free on YouTube. So I have like a bioinformatic from scratch series where we kind of go, like from the basic like data collection, um, like from the Chambo database for like medicinal compounds, and then we pretty much like prepare the data and then build a machine learning model, explore the data as well, and then finally we built like a data application. And then yeah, I also tweet sometimes on Twitter, and also LinkedIn. Feel free to connect with me there. So I really like this quote, which resonate a lot with me, and that is saying that I will argue that computational thinking and computational methods are so central to the quest of understanding life that today, essentially, like all biology is computational biology. And the reason why I really like this was that we could think of biology as practically generating, like huge amounts of data. And this is ever more becoming true in the age of AI and also like the post-genomic era and the omics era that we are familiar with. So you could check out this excellent quote from this original article by Florian in the PLOS Biology. All biology is computational biology. So this is my personal journey, which I think might give you like some perspective on how I actually got into this area of machine learning, AI, drug discovery, and how I made the unconventional path of going from biology to drug discovery, and then to working in tech. So it's quite unconventional. And so my journey started back in 2002 when I got my bachelor's in biological science. So in my third year, I was like, no, actually my fourth year, I was thinking of potentially changing majors to computer science. But then I figured like I only have like a few quarters left. So I might as well, just graduate with a biology degree. So that led me to the path of pursuing a PhD study. So as a undergrad graduate, I had no idea what I wanted to do aside from like being a medical doctor at the time. And then like I found this pretty cool documentary. I guess it was like on Discover Channel, and then that led me to the path of pursuing further studies for a doctoral degree um, in machine learning and quantum chemistry. You might know of it as like computational chemistry as well. Um, and so at the time, like a lot of the work that I was doing was pretty much simulating, generating models at a time when it was pretty much unconventional in a traditional laboratory to, like model data. Back at the time, not very much people were, I mean, people were reluctant to even embrace computational studies. They were more into like generating wet lab experiment, and that was like the conventional norm. It was like back in like 2002 when I started my PhD. So yeah, so it was pretty much grinding throughout those period, and then I was in academia like pretty much for almost 20 years of my life, 19 years, like 15 years of post PhD and then 4 years of PhD. So that's like 19 years. So it's practically about half of my life at the time. Yeah. Yeah. So before starting to explain each of this, so there are three parallel paths that I was doing. Like one path is like research, and then the second path is content creation or teaching. if you want to put it into the same bucket, teaching. And then the third category is coding. So yeah, so research was started back when I was like, I don't even know how to code aside from making websites, HTML, CSS. So yeah, I was using like MATLAB, a little bit of MATLAB, and also like predominantly using no code solution like the Weka software, which is pretty great. And it allowed me to like do research in using machine learning even without coding skill, and then making discovery on like DNA splice function, site prediction, predicting the biological activity of compounds and protein functions. Um, yeah, so I was teaching pretty much since 2006. After I graduated, I became a lecturer, and then pretty much like climbed the career ladder in academia, and then eventually became a professor in 2018. And yeah, so 2019, I embraced like kind of like experimented with YouTube, creating content tutorials there. Like before I go to sleep, like 11 o'clock at night, I would just record a video and go to sleep. And surprisingly, people found it useful. So, and I also found it very fulfilling, like learning from the community. Viewers are, like providing feedbacks and pretty much allowing me to learn from this fun endeavor. And also in parallel, coding pretty much started after 2003. I think it was sometime, yeah, it was like 2011 where I first learned Python, and then R, develop models, both in R and Python. And then I started to experimented with data applications, uh, probably in 2016. I tried to submit a paper to Nature Biotech. We pretty much like compiled a large data set of green fluorescent protein. It passed the first editorial review, and then it went to peer review, which was pretty interesting, and a good learning journey as well. So unfortunately, like all of the reviewer found it interesting, but then their only concern was the data app. I mean, the website was built using Joomla, which was like a content management system, and it wasn't very interactive. You can't build models. You can't perform interactive data visualization. So unfortunately, it was rejected. And so that led me to the path of figuring out how could I learn to build interactive websites. And yeah, so my journey started actually with Shiny in R. And then that was for pretty much like the first 2 years. And then one of my subscribers suggested that there was a new library called Streamlit for Python. And so I created this tutorial video on my YouTube channel. And that pretty much led to several other tutorial videos as well. So all of these journey are allowing me to learn more into coding and the interface with biology and machine learning. When I was in academia, I was doing bioinformatic work, and it's pretty much similar to data science, where you pretty much replace the domain knowledge with biology, and then you apply statistics and computer science to make sense of biological data. Yeah, so I drew this infographic, illustration to pretty much summarize what are some of the common tasks in bioinformatics. And it pretty much could be categorized into search, compare, model, integrate, and curate. Yeah, so like for example, you want to search for a protein or a gene from a database, and then you want to compare it to others through sequence alignment or structural alignment. You want to build models. You have sequence that might not have a structural model, and then you want to predict it. You have compounds where you want to find out how well it performed in inhibiting a protein. Then you want to build models out of it. And also pretty much to integrate, curate data, because once you generate a lot of data, you have to pretty much clean and maintain the data. And predictive modeling, this small niche area, is what I was doing for the past 19 years. So yeah, you already know that, like after the Human Genome Project, it was fully completed in 2003. After that, it pretty much unleashed a whole new frontier of other omic, like proteomic, transcriptomic, lipidomic, glycomic, metabolomic, interactomic. Pretty much figure out like the various space of all of the interactions between protein ligand, ligand interaction, and it has wide implication for the life sciences. Yeah, so if you were to pretty much summarize the bioinformatic pipeline, you could essentially break it down into three essential pieces here. So you have biological data generation, you have sequencing, uh, next gen sequencing, you have protein structure elucidation. Now we have AlphaFold for predicting structures. You have gene expression, and then you want to analyze that data, right? You generate the data, but then you can't discern the pattern just by looking at it. You have to analyze it, summarize it, aggregate it, and then you're applying algorithms, statistics, data visualization, machine learning to make sense of the data. And then, most of the papers stop there, right? Most of the research articles just pretty much stop with model generation, prediction, and then report it in a static form in a table, tabular or like graphical plots. But then they're not interactive, right? You publish models and they're in the paper. You see that it has good prediction performance, and that's pretty much it. So I think it was in 2016 or so, I had a chat with Professor Brown. He was visiting Thailand. He was from Michigan State University, and he's doing a lot of open research. So he shares pretty much his findings in GitHub repository. He shares all of his research code pipeline in his GitHub account. And then with that pretty much spark, this thing with me that, okay, when I'm doing research, let's make it open. Let's share all of the data. And then I kind of learn about GitHub, version control, and pretty much like sharing all of the data that we accumulated in our research, and then share also the code so that others could, make use of it and build upon that. And then Shiny kind of came afterwards. Not really sure when, 2017 or so. And then, yeah, Streamlit came about 2018, where I pretty much learned, and then, build data apps from then. So you see the progression here. You have data accumulating in databases, and then you make use of those data, applying. You build machine learning models, you generate algorithms, or you make data visualization, and then if you want to make it interactive, you want to make it so that other researchers, other biologists, other chemists could make use of it. It would be great if you could share the model, as a living model where others could pretty much give their input and then make prediction, and so that they could use it in their own research instead of having to reproduce the algorithm mentioned in the research article. And then, that could be another rabbit hole where they might or may not, they may or may not reproduce the model, and they may or may not make use of the model for making prediction. So if you share it as an interactive website and a web application or data app, you can be sure that users will be able to, make the most use of your findings, right? Because you spent several years or several months in developing your research, only to be published in a paper that is, it might or might not be read by others. But if you have it as a shareable form where others could make predictions with it, it will be more impactful. Yeah, so the landscape of bioinformatic and also its challenges has pretty much could be summarized into the following challenges here. So we know that data is accumulating at breakthrough speed, right? We have large volumes of data. And then we have heterogeneous types of data and different formats. And that pretty much is limiting our ability to make use of all of the data. And then how about the usability? It might be difficult for others to reproduce your work. It might be difficult for others to, take the data that you mentioned and you developed, accumulated, and then build a model out of that in order to reproduce the model mentioned in the paper. And that would influence the adoption rates, right? Because static tables and charts are not usable by the end user or researcher. And then you have also, like the reproducibility issue, because people are using different operating system, but then we now have containers, right? We have Docker container version control, and so that is no longer an issue. And so all of these are challenges and pretty much guidelines for how we could embrace open research. Because, if everyone share their data, share their resources that they created, we don't have to work in parallel silo. And then we could make this kind of like a research snowball effect where one research builds upon the other. And if all of their scientists could contribute to this shared pool of resource knowledge, then I think, bioinformatic progress will move even more faster. So if I could summarize like the TLDR of what I've been doing for the past 19 years, it could be summarized into this cartoon illustration where essentially you draw an input molecule, right? You provide an input molecule where you don't have idea on its biological activity, and then you put it into a machine learning model, and then the model will make a prediction on the likelihood of it having a predicted bioactivity of, for example, a log PSC 50 of 6.5. So that's the essence of what I've been doing. And to the title of this talk, connecting the dots between theory and application. So we know that theory is pretty much vague, right? It really is vague in the terms that when we read articles, we have to interpret, we have to reproduce it. And so therefore there's like this bridge. If we were to share the data, the app, the code, it will allow you to make use of your findings. So pretty much building this bridge between theory and application and utilization. And so, yeah. So I've been doing that by building data apps. And having an interactive data apps pretty much allows you to have conceptual, ideas or theory come alive through, like interactive exploration. So now researchers could do many things, right? Using these interactive apps. They could design molecular models. They could initiate, like protein folding simulation experiment. They could visualize how protein are binding to other ligands. They could visualize how mutations to specific region of the protein would influence the binding. They could visualize how, like a library of compounds bind to a target protein or a family of target proteins. And so these could pretty much turn abstract, ideas into more intuitive and more insightful discovery. So I'll provide a couple of ideas and examples. Actually, this is the wrong URL. It's actually abtesting.replit.app. So this is an example of a Streamlit app where you adjust the parameters here, and then you look at the effect on the sample size for this A/B testing tool, right? Because it's often difficult to pretty much see how different parameters affect the statistical properties. So if you just adjust the slider here, you'll be able to see it in real time, how the property is affected by the parameter. Another example is this is for a machine learning model to predict the penguin species. For here, you could allow the user to play around with the features slider here. You slide the features, and then you get to see how it affects the prediction of the categorization of the 3 different types of penguins here. So you'll be able to see in real time, which feature influenced the categories. And you could play with this through this interactive data app. You go to palmer-penguin.streamlit.app. And yeah, I think this was like pretty much like the last research article that I've published with my colleague before I left academia. So it was like 2001, and then we submit the paper. I think it was like 2002. So yeah, I left academia in 2021. And in 2022, we published this article. So it's pretty much a quantitative structure activity relationship study where you try to find correlation between the structure of the compounds and then try to find the relationship between the biological activity, which is the PARP1 inhibitory activity. You could play with this interactive data app through this link, parp1pred.streamlit.app. And you could reproduce this. I mean, the full code of this is shared in the top right-hand corner if you go to this application. And then you could, replace it with your own data, and then you could make a prediction app out of that. Another example is an interactive dashboard. Like you could use a spreadsheet, and then you could, try to manually look at how the different year affected the migration over time of the different state in the US. Or you could develop an interactive dashboard where the user could play with this, pretty real-time dashboard and see how that affects their population migration. And so if you were to ask, how could one go about building a data app? Well, firstly, you have to decide on a data problem that you want to solve. And then you want to build a workflow in a Jupyter notebook because it allows you to explore the data and then find relationships between the variables and then figure out which algorithms to use for building the machine learning model. Yeah. And then you'll decide on a web framework to use, either RShiny or Streamlit for Python. And then you want to install the packages or library, and then you will go about building the data app. Yeah. So the problem example that I mentioned was this PARP1 pred web server that we're building for the PARP1 protein. So it's responsible for DNA double-strand breakage in cancer cell, which will cause cancer. So we're trying to inhibit that. And so the data set was pretty much an imbalanced data set of 1720 active compounds. Compounds that are able to inhibit the PARP1, and the inactive compounds that are not able to inhibit the protein. And so we applied this, imbalance pre-processing in order to make it robust for the prediction. We use for Python libraries, we use Streamlit for the web framework. We use Pandas for the data wrangling, loading in the data and filtering the data. We use RDKit for representing the chemical structure, and then we use PaddlePy for calculating the molecular fingerprints. We built the entire Python workflow in a Jupyter notebook using Google Colab. So that encompasses, performing exploratory data analysis and building the machine learning model, and also data visualization. And then we decided on the web framework, Streamlit. So that was before I joined Streamlit, actually. So it was, we used Streamlit to build out the data app, and, to install it, you just go to pip install Streamlit. And so Streamlit apps could be built, essentially in 2 lines. If you want to build a simple Hello World app, you just import the library and then you just use st.write to build the app. Obviously, to make an interactive data app for bioinformatics, you'll copy the entire workflow from the Jupyter notebook, and then you put it into a script file, and then you name it Streamlit app.py. It could be other name as well. And essentially, you put in your workflow in here, and then you, instead of using, like native IPython tools, you'll be using Streamlit widget, which will allow you to, create text box and submit button. And there are third-party components where you could also add like a chemical structure drawing widget. So user could just draw the chemical structure in there. And there are ample, third-party component where you could also add like a molecular viewer, a protein visualizer, for example. And because the app is interactive, users could make prediction in real time, and developers could be researchers. So you don't need to hire a developer to create your application. So if you already know Python, if you already know how to create your Python workflow, like you'll be able to build this data app in no time. So the code of this bioactivity prediction app is shared on GitHub. So if you go to github.com/dataprofessor/PARP1, you'll be able to have access to the code. If you go to PARP1pred .streamlit.app, you'll be able to have access to the demo app here. And yeah, we actually created a extensive book chapter published by Elsevier on so I think it's Academic Press, building bioinformatics web application with Streamlit, and we did that with several of our collaborators who all of which are in academia, and they're also using Streamlit for building their data apps. And I believe that's all we have for today.